So the last experiment which I want to present to you, I have put into 3 parts. And the name of this experiment is detection of DLK1 expression. You know already from my presentation what is DLK1, what is it good for, what we have identified. So you know that this DLK1 is regulated by Wnt signaling, and it plays an important role in the adipogenesis, so differentiation of adipocytes. What show our not yet published data is that T7 and SKNC15 both have the ability to regulate the amounts of DLK1, and by 2 different mechanisms. The first one is transcriptional regulation. That means that T7 can specifically affect the amounts of DLK1 transcription, the intensity of transcription, thereby the amounts of DLK1 mRNA, which is in particular organism, tissues, cells expressed. In our experiment, what we use for this practical course are mouse embryonic fibroblasts, which are isolated either from wild-type mice or from knockout mice. In this case, what I have used was, on one hand, MEF cells from wild-type and T7 knockout, so not double knockout, but only single knockout. And the goal was to identify the levels of expression of the DLK1 gene. The first step for this protocol is the isolation of RNA. So this is the first movie which I show you now. Here we have the 2 plates. As I told you, these are so-called wild type, so T7++, SKNC15++. Important was also to know that it is clone number 3, because these mouse embryonic fibroblasts were isolated from different embryos, passage number, and this was freezing number in our database. Cells were prepared on 16th, and here you have another plate, which was the knockout for T7. And then we are coming to the protocol. So the protocol I send you, this was using a kit, which is an advantage because it's a reproducible quality. The principle of this isolation is the Komchinsky-Sachi technique, where you isolate either DNA, RNA, or protein. And the advantage of this particular system is that you don't work with phenol chloroform, so it's environmentally friendly and not dangerous. First step is to trypsinize cells. So cells, living cells in culture, have to be really confluent. And then what you have done yourself in the experimental work at the beginning of the course, you trypsinize cells, collect them in the full medium, then centrifuge down in order to separate the medium from your cells. And then the cell pellet is lysed with a specific lysis buffer from the kit. So going to the next step. You see here these 2 examples, sample from wild type cells, from knockout cells. The size of the pellet here is a little bit smaller, so that would affect the final yield of the RNA. That is always important, as I showed you, to have a look first on the cells under the microscope, if they are healthy, if they are not contaminated. In this case, you have to see what is your starting material, that you have enough of the cell pellet, the size of the cell pellet, what you can expect. Secondly, here you still see the medium, which has to be separated from the cell pellet. So this has to be removed by pipetting, because otherwise the sensitivity of the technique is not high enough. And then the next step is adding the lysis buffer from the kit. And you see that this is disquality thionate, which is the technique Konchinski-Sachi. After lysis, the cell pellet turns into a turbid solution. This is the kit, so I'm not advertising this particular product, but it is only for your information. What would you use if you do this experiment? We are starting with the first step with these columns. What is important and necessary always to label everything and be careful, because ethanol is removing the labeling. If you are working with many samples, it's dangerous. Then the next step is pipette lysate on the center of the sieve. So here you have to be careful when it's a small volume to go directly on the bottom of these columns, not on the side wall, because then you lose a lot of the sample. So then it's several centrifugation steps all done in the microfuge. So it's fairly fast protocol. Important is to keep the proper speed according to the protocol and to go step by step by washing and afterwards elution. So washing is with ethanol, 70%. There you have to be careful that it's really 70% ethanol in order not to precipitate proteins, which was completely clogged the filter. And the last step is the collection into fresh tubes. So there is washing after these washing steps. Here is important, in the kit you receive the buffer without ethanol, so that has to be added and marked. Don't forget to do that in the practical life. The exact protocol you have already received, so you can go then one step by the other and have a look. There are different washing steps. Centrifugation, collection, removing ethanol from the sample, and the last is elution with RNase-free water. So here you have to be very careful because we are working with RNA. Naturally, you don't want to contaminate it with RNases. That's the reason why there is a special treated water part of the kit. So use only this, and also all plasticware has to be clean, free of RNases. Always wear gloves in all steps, and don't even try to talk about above the samples, because you can also contaminate it with saliva. Elution, once again, label properly your tubes, because this will be saved for the next step, which is the cDNA. And when it is stored at -20, you can store the RNA for a longer time. So it's a very useful technique, because if everything is done well, then the yield is big enough for several repeats of the experiment by PCR detection, qPCR detection of your genes of interest. Here is this step important. It's very small volume, so this 50 microliters is more or less one drop of water has to be directly on the center of this sieve, because otherwise you will not elute it properly from the column. Centrifugation is repeated either once or twice depends on the yield. So if it's repeated twice, the yield is slightly higher. The second elution is with smaller volume, and you collect all the material which was eluted. And then comes the important step, and that is the determination of the, on one hand, quality, on second, the concentration of your RNA. So this instrument is called nano drop, because there you use just one single drop of your material. With this instrument, you can measure the concentration of DNA, RNA, proteins, and so on. In this case, we have selected RNA. That's the software program for the RNA measurement. First, you have to start with the blank. Blanking solution in this case is water, because we have eluted RNA with water. And here you see the very small drop, which is approximately 1.5 microliters of solution, which you put on this location in the instrument to start with the blanking. Then the measurement is performed, and here is the result. So what you can see, one important parameter is the ratio in adsorbance at 260 nanometers divided by 280 nanometers. And there you should know for the RNA is important to have the value around or above 2, then the quality is good. The second number here is the concentration of RNA in your sample. So since we have selected algorithm for RNA, also the quantification is already performed for RNA, and there you see 609 nanograms per microliter, because the factor for RNA is 40, so it knows exactly how to calculate the concentration. So this is per microliter, and you know that we had 2 elution steps, so the yield was pretty high. That was the first sample. Second was the knockout, which was lower. The reason for that is that, as you have seen at the beginning of this video, the pellet for T7 knockout MEVs was smaller than for the wild type, and that's probably the reason why we have also slightly lower yield. The quality of the sample is exactly good as the first one, so this is very important because we don't have degradation, and that gives us a good chance for further parts of the experiment for the determination of expression of the gene of interest. So that was the first part of the experiment, that was the RNA isolation. So the next step is the synthesis of cDNA, once again using a kit. Important is not the manufacturer, but what are the components of the kit. The most important one is the reverse transcriptase enzyme. Then you get buffers. Then you have DNTPs. In this case, it's a mixture of all 4, and an RNase inhibitor, which also protects the RNA. Primers, there are 2 options. You can use either hexamer primer, which is less specific but gives a better yield, or oligo-DT primer, which is more specific for mRNA. And water has to be naturally ultra pure, meaning not containing RNases. Important is how to protect your samples. So these are our RNA samples, which you have seen before, kept on in a special metallic rack, which keeps the homogeneous temperature around 4 degrees. Then we have so wild type and T7 knockout. Here is prepared special PCR tube with the cap, which is used for 8 samples. We will use only 2 of them. Important is to be careful while handling these samples. Always put it on ice. Don't touch it with bare hands, only with clean gloves. Here are parts of the kit. So water, the NTPs, the mixture, primers, which we'll use, and the buffer, they are kept at 4 degrees. So on ice, the whole kit at -20, but when you thaw it, then put it on ice. And two parts of the kit, mainly RNase inhibitor and reverse transcriptase, are kept at -20 because this is an enzyme which is very sensitive to higher temperatures. So you always have to remove it from the freezer for a very short time, just add to your samples and put back. And if you need to have it on your bench, then always keep it in this special rack, which is cooling down to -20 degrees. And then you can be sure that your enzyme is not degraded, that it will properly work. So always start pipetting with the highest volume, which is in our case water. It has to be calculated depending on the concentration of RNA. So you know already concentrations, which we have measured at the end of the previous video. And the total volume should be 11.5 microliters. So this is the concentration of RNA for the first sample if you want to use 1 microgram. So the reason why I have measured the RNA concentration was that for this step, we want to use exactly same amount of RNA. So for wild type, it's this volume, and for the knockout, it's a different one depending on their concentration. So we start with water. It's this special PCR grade water, not containing any RNases. You put it into these PCR tubes, and then the next step is to add the samples. So this is the sample of RNA. Next is DNTPs, also from the kit. So it's a mixture of all 4 DNTPs. Then RNase inhibitor. One important fact is you have to always use fresh tips, ideally so-called filter tips, in order not to contaminate your probes one by the other, because that would ruin the result of the experiment. So RNase inhibitor is a very small volume, 0.5 microliter added to each sample. Then the hexamer primers. Buffer containing all components necessary for the reaction. So it's a fairly simple experiment. Important is only to pipette accurately and not to contaminate samples. And as the very last step, you add the enzyme reverse transcriptase, which has to be kept at -20 degrees. Then next step is to centrifuge samples down, because it's very small volume. So if the reaction has to run properly, you have to collect all samples at the bottom of the tube. And then we come to the PCR machine. Where is this program? You have it also in the protocol written. So the first step is to activate the enzyme at 30 degrees, 10 minutes. And then we go to next step, which is 60 degrees for 52 degrees for 60 minutes. So 1 hour. And depending on the type of reverse transcriptase, there are different temperatures. So for this particular one, it's 52 degrees. And then comes the last step where you deactivate the enzyme by basically boiling the sample. So you destroy the protein by 99 degrees for 5 minutes. And you have only cDNA left. And afterwards, it's kept at 4 degrees until the next step comes, which is the qPCR. So it's fairly simple, but it has to be properly done. That means that your samples which were centrifuged are kept at 4 degrees, then you put it into the machine which is already pre-warmed, close it, close the lid, and then since we have very small volume, it's important that it's also done with a preheated lid. So you run the program for reverse transcription. You have seen the algorithm, and then you have to select the volume which is used, volume of samples. This is all necessary for proper heating. And then you see that the lid temperature has to reach 99 degrees, and then the whole reaction starts. Why we are using the heated lid? It would evaporate the whole sample to the top or to the lid, and we would lose the sample. So if it's pre-warmed lid, then you don't lose the sample, and then you have better efficiency. Is this clear, or do you need any further explanation for this step?
That's clear.
Okay, then I will close this movie and go to the last one, which is the PCR. So this is the last part of the experiment. And what we want to detect is using so-called Tachmann system, the Tachmann probes. You can have a look and figure out how does it work. And that can be part of your protocol to a brief information about the Tachmann, because there are several options how real-time PCR can be done. So one of them is using CyberGreen. What we are using here is so-called Tachmann probe. So the system, you have to understand how it works. It's important for the whole experiment, and that can be part of your protocol. The advantage of this system is that whatever you want to detect, any new gene, you always use the same algorithm. So it's always same temperature, timing, and everything in the PCR reaction. And that's a big advantage because you don't have to establish for each gene a new protocol. The disadvantage is naturally the price. But it's a very accurate technique. What we wanted to detect in this case is the expression of the gene DLK1, and it has to be normalized. So that means that we have used same amount of RNA to generate the cDNA, but we don't know how much from this sample represents our gene of interest, in this case DLK1, and how much we have of different cDNAs, so it has to be normalized on a housekeeping gene. Therefore, we are doing two measurements. One is the Tachmann detection of DLK1, and the second one is the detection of the housekeeping gene, which is GAPDH. And then we will normalize the results of DLK1 on the GAPDH in each sample and compare the differences in expression of DLK1 between wild type and T7 knockout MEFs. So once again is using this technique a kit. So we have a gene expression master mix. The largest volume is 5 microliters. We have triplicates for each sample, so 3 times wild type, 3 times single knockout material. But because it's small volume, I have written here it's for 7 samples to have some reserve. And you mix this gene expression master mix with the specific probe, Tachmann probe for your gene of interest. So it will be 7 times for DLK1 and 7 times for GAPDH. Has to be kept on ice. And then we put this 35 into the master mix tube, mix it with the specific probe. So you can order ready-made Tachmann probes, this case in DLK1. Mix it together and then pipette into the specific PCR tubes. The second one is the gap DH. So here are the master mixes. Then you put master mix into the tubes, and for this we have a very useful device for this particular instrument where you can always turn on a light for that sample where you have to pipette, since there are many samples and it's easy to make a mistake. So we go one by one, and first you have to fill in everywhere the master mix, and the second step is to add your samples. So what I did was 3 times wild type, 3 times T7 knockout. The first row was for DLK1, and then once again 3 times wild type, 3 times knockout, and the second row was for gap DH. So always one sample, and then you go to the next. First is the master mix for DLK1, 5.5 microliters, and the second is for gap DH, always 5.5, that's the master mix. And then our samples, cDNA, which you have seen in the previous video, how they were generated. And there we have used 4.5 microliters. Then the next step, you cover the whole PCR tubes with a special foil, which is very important to seal it properly. That means that you should have here a very good contact between the top of your PCR tubes and the foil, because the temperature is very high, so it runs up to 90 degrees, 99 degrees, and it would evaporate. So you have to be really very careful that you have enough material of this foil that is completely covering your sample. So our samples are these 12 samples here on the top. That was the reason why I left bigger leftover. It would not be good here where you don't have such a good contact. You can lose these samples. And then we have a small plastic device with that we seal it properly several times. So on one hand, it has to be closed. On the second, it's very important that it's really clean, because during the reaction, it will be the instrument, the PCR device, reading the signal from this probe, which is a light signal. And if you have some dirt here, that would affect the result. So with this device, I sealed the PCR tubes, then centrifuged it down, because it's very small volume. You have to centrifuge it in order to collect your samples. So short time, but it has to be properly done. And then we go to the PCR machine. So this is a special PCR device for real-time PCR, where you can pre-program your protocol. In this case, it's 50 degrees, 95 degrees to activate the enzyme. Then the nature samples. Then the annealing step is 60 degrees for 1 minute. During this step, 60 degrees, is the signal collected and measured. This is important to know which dye is used in your samples, because there are several different possibilities. So we have in our Tachmann probes FAM dye, and then at the end, after 40 cycles, is cooling down to the room temperature, and then you can read out the results. So all results are collected on a USB stick. Here you see once again our protocol: pre-warming, heating up, activating the enzyme, and then these 40 cycles. During this cycle, always is the reading of the signal. You open the instrument, put in your samples, they have to be properly sealed, clean, close it, and start the run. The whole procedure is fairly fast, so it takes about 1 hour something to do the whole PCR reaction. And then comes the next step, which is the analysis of data. For that, we have to use the software for this instrument, which is shown here. I will show you few parameters which are important. So this is our read. This was our reaction, which you have to select. And then what we are interested in are these CQ values. You have to select your samples where are in the plate. So we had 6 and 6 samples. The upper part is the DLK1, lower part is GAPDH, left 3 are wild type, right 3 are T7 knockouts. Now, what we see on the next image is the curve. So you see the number of cycles necessary in order to get some signal. So at the beginning, as I told you, after each cycle or during each cycle, the 60 degrees is the readout performed. There is no signal. And then here starts some signal. Since we have now selected all samples, you see the signal for all values, all samples in the same graph. And here you can already notice something interesting. So you see 3 curves which are very similar to each other. This is a good sign because these are samples, triplicates of the same sample, and they should be like this. They should be very, very close to each other. And then you see another 3. So these 6 curves belong to the same gene, and that is the GAPDH. Naturally, the quality is good because you have seen already the readouts of the RNA concentration measurement, but it is also important to pipette properly, so they have to be almost all identical, these lines. And then there is a small difference because although we have used the same total amount of RNA, most probably it's not completely identical or not accurate enough. That's the small difference. And then you see another 3 and 3 curves, which represent this DLK1 signal, and there is a huge difference. So one sample has a detectable signal already after 22, 23 cycles, and the other one much, much, much later, which means that in this sample we have high amounts of the DLK1 gene, and the other one we have much lower. These ones are the knockouts and these are the wild types, but we will go to the details in the next pictures. Important is that we see also saturation. So after a certain number of cycles, the curves reach a plateau. And this is a logarithmic image where you see first few cycles where is no specific signal. So this is all unspecific signal. And then comes a nice exponential curve, which you see here. And what you can do either automatically or manually, you can put this threshold in the middle of your exponential part of the curve. And then you look up on the differences in the so-called CT values between samples. So where the same intensity of the signal is reached after how many cycles, and this gives you the difference between samples, between the amount of the product in different samples. So here, for instance, we have 3, so triplicates of the wild type cDNA, we are detecting the LK1 gene and the number of cycles necessary to reach this level of 1 is 34 cycles, 34, 34. So it's almost identical, which says that the quality of the experiment is good because triplicates are reproducible. Number of cycles is very high. That means the amount of our product is very low. So this is DLK1 for the wild type MEFs from wild type mouse. Logarithmic presentation, you see this is the background, and here comes the specific signal. Lines are more or less identical. Second part is the sample TIS7 knockout MEFs, detection of DLK1, and number of cycles necessary to reach the same intensity of signal. So it's 17, 17, 17, good pipetting, almost identical values, and much lower number of cycles necessary to reach the same intensity as for the wild type. So here we have much more of the same material of this product, which is DLK1. And if you look on the curves, you can nicely see that here. So we have identical curves and then reaching plateau. Next sample is GAP-DH. First is wild type, triplicate, identical number of cycles, so good quality. And number is 19, which is interesting to compare then with the knockouts. So you see in the middle of the exponential curve is this threshold, and that was around 19 cycles. And when we go to the next sample, which is in this case the triplicates for this 7 knockout material, GAP-DH, and here you have the CQ. It's lower, so there was more material, more cDNA. Once again, curves have very similar pattern. So the quality of triplicates is good, but we have much lower CQ than for the wild type. Instead of 19, it's only 13, which tells us what? That we have more material. That means we have to normalize the signal for the DLK1 on this amount of material which was used, and that helps us to normalize the result. And here you see 6 lines for the whole, so that was 13 and 13, and this is for the DLK1, and there you see 33 versus 17, so there is the big difference. Now we are coming to the end of the quantification. You can export this data in Excel format and then do the quantification itself. So you have seen in this movie how the measurement was done, and I have sent you files in Excel format where you have the results. I have simplified everything in order to make the task for you easier. So all what you have there are the numbers of these CT values for each gene, all samples. What you have to do is to follow the protocol, the formula which is written in the protocol, and make the quantification using this formula, which is this delta CQ, and then prepare a graph. Everything is explained in the protocol. I will try to open it now and show you once again. So important is what is written here. I have written all details there. So what you do is to do first, this quantification is called delta CQ. Delta CQ is CQ of the target gene. In our case, target gene we call VLK1 measurement, minus CQ of the housekeeping gene, which is GAPDH. And you do the quantification for each measurement. So we have six values, six measurements, 3 times wild type, 3 times T7 knockout. Next step, normalize the wild type. It's called to the wild type. So you first normalize to the housekeeping gene and then to the wild type. So what you want to know is the difference between your target group. In our case, it's the T7 knockout maps, and wild type is wild type. So this calculation is called delta CQ, where you deduct from delta CQ for the target group of T7 a mean, so the average value of delta CQ of the wild type. So that means that you have calculated this first, and you have 3 values for delta CQ for your wild type group, and that is always DLK1 minus GAPDH. Then you calculate a mean for this wild type group, and you deduct this mean from each single result of your target group. And then when you have done that, you make the exponential expression. So that's the logarithm to the minus delta CQ. You have then 6 values, because you do this measurement, this calculation also for your wild type sample. So it's wild type minus mean of wild type. And it's this 7 knockout minus mean of wild type. And then these 6 values, you have to calculate an average and standard deviation, and then make the graph. So we have wild type set up as number 1 and compared the knockouts. In this case, this is just an example of the graph. It was done for double knockouts. Now in this experiment this year, we don't use double knockout, but a single knockout. And then you calculate how is the difference comparing T7 single knockout to the wild type. Is it lower? Is it higher? How much is the fold increase in the single knockout? So this is your goal. What you have to do, you have the Excel sheets where are the measurements. You have to find out what is what, and then make this calculation, and then the final graph, which will be properly labeled. So you have to write down which gene was detected, normalized on what is your wild type, what is your target group, and make this type of graph as if it would be used for a publication. And then you can write a short discussion explaining what is the conclusion from this experiment, what we have used, which samples, and what is the effect of a knockout of T7 on the expression of DLK1. Is this clear?
I have a question.
Yeah.
Because I already calculated it, and I have for the T7 knockout a fold change of about 76,000. So if I want to do a graph, I can't see the wild type anymore because the wild type is around 1.
So there you did a mistake, because it cannot be 76,000, because I did the calculation as well using this formula, and the difference was, I think, 2.7 fold.
But if I have the...
Using these numbers which were there, 1911, I can open it now, just a second. Yeah, So it was 2.79 was the mean, and standard deviation was 0.005. So do it carefully, because I have really used this formula which I'm showing to you. I didn't change anything.
Yeah, I also didn't change anything, so I don't know what I did wrong.
You have to go carefully once again through your calculation. There must be somewhere a mistake. I don't want to show you my results, because this is your job. You have to do it. You must learn it. But the numbers
I maybe show you mine, or
numbers are correct, and calculation as well. So just carefully have a look once again on your calculation.
Can I also have a question? Can I also? Question. For the first point, so normalized to the housekeeping gene, delta CQ, and so on. So we have different values, 1, 2, 3, 4, 5, 6, and we just divide this through the gap DH. So we always take 1 and 1 and 2 and 2 and 3 and 3.
Exactly.
Right?
So you do it the first sample, and for the DLK1.
Yes.
Minus first sample for gap DH, second sample for DLK1, minus second sample for gap DH, and so on.
Okay, and then you have the delta CQ expression 6 times, and you always have then minus the mean of the wild type. Yeah, okay.
Exactly. So we have 6 values.
Yeah.
And you have 3 values. So it's the mean of wild type only.
You have 6 values, and you
yeah, because you have 6 individual values for 3 times wild type, 3 times T7 knockout. But for wild type are only 3 measurements. So the mean of the delta CQ expression for wild type is from 3 from the control group.
Yeah. The mean is from 3 values, and you have 6 values that you have.
So then you go, the first value doesn't matter if it's wild type or this knockout, but it's the first minus mean of wild type. And the second and third and so on, and always deduct this mean of wild type.
Yeah. And then you have a delta CQ expression 6 times. And for the diagram, we have two graphs. So we have then
3.
again.
No, you have 3 and 3. So you have 3 values.
Yes, 3 and 3.
For wild type and 3 for double knockout.
And you have then the mean for the graphs in the diagram.
Exactly. So mean of the first group, which is the wild type, and mean of the double knockout.
And then we can normalize it to the MF wild type, or is it 1? Or has the result to be 1 for the mean of the...
Yeah.
What is yes? So it has to be 1.
1 is the mean, and you have 3 values. So you take the mean as number 1, and then you compare the first value against this one, against the mean. Because otherwise you cannot get the standard deviation for the wild type. So you have to compare always each value against the mean.
Yeah, but for the third step, then you have the delta Q expression. You have 3 values and 3 values, then you have the mean. But is the mean value of this delta Q expression for the wild type, has it to be 1, or have we normalized it again to 1? Because in the graph there's a MNR level 1 for the MF wild type.
So you calculate the mean for the wild type.
Yes.
And then you say that this represents 1.
Yeah, okay.
And then you compare each single value of the wild type, so all 3, against that one, which is the mean, which represents 1. So then your individual values are larger, higher, or lower than 1.
Okay.
And then you compare the same way also the knockout values against the wild type mean, which is 1. Is it clear?
I think I am at the next step, but I just will try it, and if it don't work, I can write you an email.
Yeah, sure.
Yeah. Okay.
Okay, so I hope that it was somewhat useful to you, that you have seen the whole protocol as if it would be done in your hands. Unfortunately, I cannot offer anything more, but now you have protocols, you have results, you have seen what was done, and then I will save these videos today and put them on the YouTube and send you the link to the YouTube where you and your colleagues mainly who were not attending today's session can have a look. And now we have 21st of December. So in order to be fair, because it's naturally a little bit difficult for you, let's say until end of January, that means 30th of January, please send me your protocols via email. If you have any questions in the meantime, you can send me an email all the time and I will try to answer your questions. Itself.